On today's podcast for Inquiry, I speak with Dana Korneisel. Dana is a PhD candidate in the Madden Lab at Carleton University in Ottawa. She is studying the origins of the neck during the first vertebrate transitions to land, its adaptations across the stems of today's crowned tetrapod groups, including amphibians and archosaurs, and the developmental origins of the neck bones in modern model organisms such as alligators, axolotls, and frogs. Outside her academic work, Dana has many hobbies, including needle felting, beading, amigurumi, and paper quilling. Dana is an avid ice skater, an amateur acrobat, and a circus artist specializing in aerial fabric. In today's episode, we talk about her area of expertise, the tetrapods, and what they encompass, and within that, especially amphibians and frogs, and one of my favorite animals, the axolotls. We also talk about her children's book, Dana Digs Dinosaurs. I want to encourage everyone listening and watching to Podcast for Inquiry to please consider making a donation to the Centre for Inquiry Canada. We are an entirely volunteer-run national educational charity, and we rely on your support to continue making Podcast for Inquiry and the many other programs that support free thinking, secular values, and rational inquiry and evidence-based public policy. Without your support, none of this would be possible. Please go to centerforinquiry.ca/donate and make a donation and consider becoming a member of the Centre for Inquiry Canada. Thank you. I now bring you my conversation with Dana Korneisel. Welcome to the Podcast for Inquiry, brought to you by the Centre for Inquiry Canada, a national educational charity supporting your community for reason, compassion, and secular humanist values. You have answers, we have questions. I'm your host, Leslie Rosenblood, board member and secular chair for CFIC. Welcome. Dana Korneisel, thank you for taking the time and speaking with me today on Podcast for Inquiry.
Of course, thanks for having me.
I wanted to talk to you because you are someone who knows a lot about something that I know a little bit about, but find endlessly fascinating. But let's start with the big picture, and then we'll get down to a little bit more details. You are a PhD student at Carleton University, and broadly speaking, you study tetrapods. Is that right?
That is right, yes.
Okay, so I had to look it up. I had an idea of what tetrapod meant, and I was largely right, but what wasn't quite there. Now I have a decent idea, thanks to Wikipedia. But why don't you explain what is a tetrapod, and within that, what your area of expertise is?
Of course. So there's a big clue in the name. Tetrapod is 4 feet. That's not always accurate, but ancestrally it should be accurate for all of us. That describes pretty much any animal with bones that has at some point approached or lived on land. So fishes have bones, but are generally not terrestrial. So any of those fish descendants, including us, even including whales, including anything who at some point had 4 limbs living on land, is a tetrapod. So that unites amphibians like frogs and salamanders with mammals and reptiles and birds all together in one big group.
Wow. So that's like a majority of the animals. Like, it doesn't include arachnids like spiders, but that's like pretty much everything else that lives on the land, an animal that lives on the land.
It's most vertebrates that people think about. There are a lot of fish in this world that would resent being left out. But it's okay, a lot of.
But we did say that lives on land.
Yes. Mm-hmm.
Okay, so let's talk just a bit about whales, because they're an interesting example. You say so things that live in the sea are generally exempt. Why are whales considered tetrapods? They don't have feet at all.
So when we're talking about groups from an evolutionary point of view, we're discussing their relationships with one another. So what they're doing at any given moment doesn't really matter as far as the within evolutionary time. Whales are tetrapods. They're a mammal. They're like very much firmly nested within mammals, and mammals ancestrally are terrestrial animals. So the ancestors of whales were land dwelling, and they were not the first, probably have not been the last, but are one of many groups of tetrapods that have moved back into the water. And that's something that started very early in tetrapod evolution, pretty much as soon as we have firmly established terrestrial animals, terrestrial vertebrates. We get some that lose their limbs or reduce their limbs significantly and possibly move right back into the water right away. So this switching is super common. But since their ancestors were terrestrial, or at least had all four of their limbs in place, they still are tetrapods.
And when I learned about evolution, that was one of the things that kind of blew my mind, because the image that we have, at least in a popular sense, is the descent of man. You see like the ape, and then man becomes more and more erect, and you can see it's fairly linear. Or we hear about how there was a single cell and then multiple cells, and then we came to the sea, and then we came to the land, and then it's kind of a line. You can follow any given species and trace a single line back, and what that tells us is that pretty much throughout evolutionary history, species are trying, not consciously, but there's different reproductive strategies going in all directions, most of which fail.
Yeah.
so that okay, there's this land thing, but there's this poison of oxygen, and we had to figure out how to use that as fuel instead of it being poisonous. And then as soon as we're back on land, we're like, well, maybe we should go back to the sea, and that failed for a lot of folks, or a lot of animals, but it actually succeeded in some cases. And then it's kind of fascinating that there isn't actually a straight line in evolution, but just everybody trying everything, and then whatever works survives to try something else in the next 100,000 generations.
Yeah, and you have to ask yourself what failure is really too, because if a lineage lasts for 100 million years, that is that a failure, or is it just something that eventually had a bit of bad luck, but was working? Any given animal is equally like well adapted, is equally as adapted to its environment as any given other animal at that time. So yeah, it's hard to say what a failure is necessarily. If you were unlucky enough to be around at the end of the Permian, did you do anything wrong, or were you just in the wrong place at the wrong time?
Right. Yeah, that's a good point. So tetrapods encompass a huge range of animals, historical and extant.
Mm-hmm.
I can't imagine that you're an expert in all of them. That's too much for one person to know. There's got to be some specialization that you have within the tetrapod. What is it? A group, family, class?
Oh God! what is the like technical taxonomic level?
Anyway, it encompasses a huge
within tetrapoda. Yeah.
It's a huge range of animals. So within that, what's your area of expertise?
So the way that I've limited my scope for the sake of my dissertation, which is something we all have to do, is like pick something specific enough that you can actually study it in like 3 to 5 years and make a few projects out of it. I have focused in on the vertebrae of the neck. So the reason that I've done that is one, I during my master's did a bones course with where we're looking at the anatomy of all of the boned animals and trying to learn as much as we could about every bone in the skeleton. And we did a little project during that course, picking something to investigate its homology. So investigate what
the homology. Can you just
yes?
What does that mean?
I got it. I'm on it. I was going.
Okay.
So homology is figuring out what bones are the same in different skeletons. So if I have an upper arm bone, if I have a humerus here, I have this one that connects my shoulder to my elbow, and a whale or a frog also has one bone here that connects their shoulder to their elbow. We can try to investigate whether it's the same bone from an evolutionary perspective, and if it is, then we can call it a homologue. It has homology. Whereas if it's some completely different thing with a crazy different evolutionary pathway, it just looks similar. Maybe we could call it an analog, or we could say it's convergent evolution. So, so yeah, sorry. So we did this little project where we picked a bone and we tried to find all of its homologues, all the things that are actually the same to it, or a feature on a bone, and where within tetrapods or within vertebrates at large does this feature occur. And at that time, I chose a bone that humans don't have anymore, or mammals don't have anymore, rather, and is present in crocodiles and other alligators, crocodiles, caimans, all of the crocodilians, and is also present in this lizard-looking animal called a tuatara that's from Oceania. So these two animals aren't super closely related. One is an archosaur. It's in the family that leads to dinosaurs and birds, as well as and crocodiles. So that the crocodilians are archosaurs. And the other one is kind of like a very, very close relative of lizards and snakes, but isn't quite in that group. So I looked at this bone that is in the neck that a lot of animals don't have anymore, and that got me interested in neck vertebrae. And then I expanded the scope of that a bit for my dissertation work, because fish do not have neck vertebrae. Their shoulder blades are directly connected to their skull. There's a bone-to-bone connection between skull bones and shoulder bones. But when these animals started to innovate and adapt to terrestriality or towards terrestriality, they lost a lot of bones that were at the back of the skull. A bunch of the bones that fish have that support their gills, we don't have those bones at all. We've lost them completely. And so did all the other tetrapods very early on, or at least reduced the size significantly. And when that happened, when we had those losses, a space eventually evolved between the skull and the shoulders. And that is characterized by these specially shaped vertebrae that eventually become our cervical series, our neck vertebra group. So I specialize in that. First, how it originally evolved, some specific lineages. So I've looked at some crocodilians and at the American alligator's development to see how its neck vertebrae develop. And I've also looked at, I have an active project going on the larger evolution of amphibian neck vertebrae. They only have one right now, but they used to have more. And then I also do a little bit of developmental work. So I've done some evo-devo, evolutionary developmental biology, where I'm looking at from embryo to young animal when it has bones, how do these specific neck vertebrae develop as well.
Okay, so that there's a lot there.
A lot of stuff.
Let's focus in on one aspect of what you said there. With amphibians, they came from fish, which had no vertebrae. The skull is connected directly to what we consider the shoulders, and then you said that amphibians had multiple vertebrae, so they must have evolved several vertebrae, and then they lost a few, and so that now they're down to just one.
Yes.
So what were the pressures that led to amphibians developing several vertebrae and then losing all but one of them? And are they going to keep, and make a prediction, are they going to keep the one vertebra that they have, or is that here to stay, at least as long as they maintain something close to their current form?
So amphibians and every other tetrapod, when they became terrestrial, we're talking about the Carboniferous. So if you know Joggins, the fossil site, that's the time period we're thinking of.
So how many millions of years ago was that?
Between 300 and 400 million years ago.
Okay.
Yeah. So this terrestrialization is happening at the end of the Devonian and into the Carboniferous. So between the age of fishes and the time where we get all our coal. So this these first tetrapods evolved two specialized neck vertebrae at least. The first one is called the Atlas, so it's like the Greek hero who holds the earth, and our Atlas is holding our skull. So you can remember it that way. It's holding up the globe that is our skull. And then the axis, it's something that the atlas turns around frequently in modern animals. In mammals in particular, in a lot of modern animals, it has a little process on it, and then the atlas is like a little loop that turns around on that process. So those two vertebrae, this atlas and axis, the very first two, right behind the skull, develop or evolve, I should say, in every tetrapod group. So we're all coming from the same starting point when it comes to those couple vertebrae. And if you look at the earliest or our best guess of what the earliest ancestors of amphibians are, it's a group called temnospondyls. There's these wonderful, like, kind of think of giant salamanders, but they are really different from salamanders. I don't want to like imply that salamanders are like a living fossil or something like that, but they're these massive, like even some of them are meters long, amphibious animals that were around for a very long time. And there's one particular group of these temnospondyls that seems to be the ancestor of amphibians. And those guys have two neck vertebrae. They have an atlas, they have an axis. When we get to the base of the amphibian tree, the axis, if it has that bump on it, like mammals still have, goes away. And the axis becomes kind of non-specialized. It doesn't really look any different from the other vertebrae. So we could kind of disqualify it as being a cervical vertebra. Whereas the first neck vertebra, the atlas, continues to have this specialized shape where it essentially has like two big cups on the front of it that cup up against the back of the skull. And that's the main thing that you look for in an atlas. If you find a bone and it has these two big, or even some now just have the one, but like two big cup-shaped surfaces on it, those are articulating up with the back of the skull and touching up against the back of the skull. So this happens a good while ago, like in the Triassic or in the Permian. And then so that's around like, and just after that biggest mass extinction we ever had. If you're referencing some points in Earth's history, it's near the end Permian. And then the modern groups of amphibians, and one group that went extinct probably, but has not been around for very long, or has not been around for a short amount of time after having been around for quite a while. So our frogs, our salamanders, our caecilians, which are not the best known of the modern amphibians, and then this other group called Elvenerpetontids that are sadly gone probably. They're my favorite cryptid. I like to believe they're still out there somewhere, but we just don't know. So those four groups all have just this one cervical vertebra left, the atlas, and many of them actually. This is a project that I have just about ready to go in for peer review. All of those groups evolve a process like this bump that was on the axis. They evolve one on their atlas, on their first vertebra, that is like points up into the base of the skull. And so I've gone down a little rabbit hole here because it's one of my active projects. But those groups, their ancestor evolves this process, and then over the course of their remaining couple million years of evolution till today, a bunch of them lose it. So the frogs start to kind of sporadically have or not have this process all over the place. They might have lost it for the sake of stabilizing their vertebral column because they often do a lot of jumping. And it's not necessarily good to have your vertebrae whipping around against each other if you're jumping and landing, especially when you're going many, many times larger than your body size. The caecilians that a lot of people don't know much about, they are sometimes called like worm snakes or something. The common names of that one are not super well known, but you might have seen them on Planet Earth. There was an episode where they showed a scene of a caecilian parent feeding its offspring with its skin. So some people have heard of it from that piece of media. that say they burrow, they live underground and they dig holes, and they use that by just moving their head right up and down and up and down. And so they, at some, like very recently in their evolution, in the like mid or late uh, Mesozoic and into like the Cenozoic, so that like around the time we lose at the end of dinosaurs and a little ways after that, they lose this process as well, or at least it becomes a lot less prominent, whereas salamanders still have it. So I did this project to see whether we can consider that process homologous, whether we can consider it something that is useful in identifying amphibians in fossils, and it seems to be. So that's the amphibian story specifically, with probably more detail than you wanted. But
No. I love learning about these things, and uh, every species is different, right? It has a unique history. So learning what makes them different, and what they have in common with others that are closely or distantly related is fascinating. Uh, I'm a geek and I just want to learn, and that's what this podcast
for sure.
is about, is getting into some of those details that really not very few people know, and hopefully spreading that knowledge
And I can
more
tell
broadly.
you that the that fourth group. So we have the three living groups. That fourth group that recently went extinct, the Albinerpetontids,
Hmm.
they have like a special extra bone. They evolve this separate bone that appears to be between their first and second vertebra, between their one atlas and their second vertebra. That's just a trunk vertebra. You probably wouldn't call it an axis. But a lot of people in the literature call this extra bone the axis. Um, and it appears to be a piece of bone that really belongs with the atlas, but is over its course of development, presumably, forming as a separate chunk that's sitting there between those two vertebrae, and could be considered very similar to the piece of bone that we have on our axis that points up through the atlas as well. So, yeah, they do all their unique things, and this is something that might be super similar to what you see in alligators, despite these being widely different animals, very, very separate from each other evolutionarily. But they might be doing kind of the same little thing
Hmm.
with one another millions of years apart. And after a long period of either not ossifying this bone at all or having it serving a different function for a long time.
So you mentioned a distinction, and I want to learn how you can tell the difference between the two, and that is if something is actually a homologue
Mm-hmm.
and has a common genetic or common ancestral source that and the example that you mentioned was the humerus connecting the shoulder to the elbow, uh, and but you can't just look at something and say, okay, that's a bone that connects the shoulder to the elbow. It must be a homologue, because you also mentioned that there's something called convergent evolution, where it's just out of the range of, I was going to say ideas, but again, that puts agency. But out of all the things that can be tried, some things tend to work, and so there's more than one approach that can lead to something that looks, in the end result, in the species that exist today, very similar.
Mm-hmm.
So how can you tell the difference between something that is a homologue and something that looks very similar but is an example of convergent evolution?
So the way to do that is to look at the fossil record. So you want to say, I've got these two animals at various points, or these two things. You could do it if you're a botanist too. It could be plants. But you've got these two fossils, or these two living animals that have a similar looking anatomy. Maybe it's a bone, maybe it's something else. We should hope it's a bone for the sake of what we're trying to ask ourselves here, about whether it's homologue. And then we'll look back through each one's evolutionary history. We look at its close relatives, because there are very, very, very few points in evolutionary history where you can actually call something an ancestor. So, like, with certainty. So we'll look at its close relatives back through time and see what anatomy they have. What does this bone look like in them? And if we can go back both of those lines all the way to the common ancestor, as it were, or something related closely to what we presume to have been the common ancestor between those two animals. And it also has this feature, this bone, and it's doing the same thing essentially in the same place. And we can track that across both evolutionary lines. Then we can say, okay, yes, this is the same bone. It comes from the same place. In the common ancestor, it was doing the same thing, or at least something similar enough. The function can definitely change. And when we talk about our shoulder, it was certainly doing something very different in the fish skull than it is for us today, but is still, a homologue, because we can look all the way back to those fishy ancestors and relatives of ours and see that that bone maintains its place. So that's what I did with this particular process. Is I saw that it's present in salamanders, and then I was helping with a developmental project and saw something that looked very, very similar in a tadpole, and at the time had only ever heard of this process as a feature of salamanders. But when I saw it in the tadpole, I went, oh, maybe this is more widespread than I was thinking it was. And at which point I started looking at the fossils, and pretty recent Sicilian ancestors had the process too. And then if you go a bit deeper in frogs, some of them have the process as well. And salamanders, consistent. They're consistent, reliable animals, and they have the process the entire time. We can respect them for that. And so I've looked back and said, okay, so even though this thing is lost frequently, I can look back in these three lines and see that the close relative or something near the time of their common ancestor does have this. And so then I can say that is actually reliably the same between all these animals. Whereas if I go deeper, if I go back to those temnospondyls that aren't considered amphibians, like in the formal sense of what these groups together, their group is, they aren't amphibians, even if they are amphibious. They have a particular anatomy in these bones, and they don't have this process. It hasn't evolved yet. There is one group of animals that has a very similar process. It is extinct, and when I say it's a group, that is not a very formal statement, because it seems to be a bunch of things that aren't necessarily closely related to each other. They're sprinkled all over the place. And for a while, we put them together in one group because they looked a little similar. But now with more recent research, they seem to actually be all over the tree. You get these small animals, these small tetrapods that also only have one cervical vertebra, among a bunch of other features, that were called microsaurs for a while. Small thing. Very creative name.
Yeah.
But these also have a process, but we don't know enough about their relationships to do what I did with the amphibians. We can't look at their close relatives all the way back and see very well, because they're kind of all over the place. So in that particular case, I could say with like a moderate certainty that there is a case of convergence between amphibians that I can like very nicely build their evolutionary history based on the fossils that we do have. And these sporadic microsaurs that don't seem to be closely related to each other but have this similar feature, I can call that a case of convergence, even though it's the same feature that is homologous in my amphibians to one another. So something like that is what you would do.
Okay, so it's not a simple or straightforward process. It involves a lot of research, a lot of comparison, and a lot of tracing back through history, at least as far as we can determine that history, and then making I guess a professional judgment based on what you can see.
Yeah, it involves a lot of looking at bones, and then in my case, I did a process called an ancestral character estimation, which is it's a method that's more common for phylogeneticists, for people who make family trees. That's their main thing, is figuring out how animals are related to each other, or organisms are related to each other. You can do this. They've come up with code essentially, and you can run through these processes to get like a statistical estimation of whether a feature was present or absent in a common ancestor. So you can also do this. I wouldn't say that that's something that just stands on its own, but when you combine something like an ancestral character estimation with an observation of all of those fossils, you can kind of put those pieces together and have like a little statistical support for what is otherwise kind of a visual piece of information, or is based on this anatomy on its own. So that, of course, requires you to use R. So highly, highly don't recommend. Not actually.
I'd like to come back to something you mentioned a little while back, and it's another one of your projects. You mentioned Evo Devo,
Mm-hmm.
and it was the development of the vertebrae in a modern day frog from tadpole to adult. I've always been fascinated just that we've got these tiny sperm and comparatively large but still tiny ova, and and when they merge, it contains all of the instructions to build these incredibly complex bodies that we humans have, but like that all animals have. Like I have complete sympathy for uh, people who say, without getting into all of the science that we have, that we must be designed because this complexity and all of these interacting systems that make up a body uh, are just way too. There's no way they could arise by chance. There must be a designer, and that's not true. But I understand how that would be people's intuition, and why that was the dominant thought process for many years, until just like basically 150 or so years ago. So can you share how vertebrae go from uh, just like a fertilized egg to an adult body in an amphibian, and guide us through that process? How does that happen?
I can do my best, but it might get jargony, so I'll try my best.
Well, if you lose me at any point, I'll prompt you. But I want to follow. Like I do want to learn, and this if this stuff was simple, it wouldn't be a mystery to me.
For
I would
sure.
guess.
So the steps as I understand them and as I've seen them. So this is like how vertebrae develop can be different in different animals. So and there are some like predominant hypotheses of vertebral development that might apply to some animals, might not apply to others, and the research is vast and frequently involves genetics, which you know I've done my share of genetics. I'm gonna try to avoid it in the future, but involves a lot of modern biology to sort these things out. So what I see when I am raising frogs, so we have both the African clawed frog, the larger variety. There are kind of two species, and one is quite small and one is big. We have the large African clawed frogs, and we also have axolotls in the lab. So when I breed frogs or axolotls, and then have their fertilized eggs developing, they go through the stages that you first see, that you're familiar with, where you have one cell, it splits to two, it splits to four, etc., 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 in a ball. Now, I'm thinking about bones, so at that level of development, I'm just like, yes, well done, keep it up.
We got a clump of cells.
Doing
Some of them
great.
are the same. Some of them are undifferentiated. Some of them are specialized. Whatever. Keep going. Keep going.
They're doing it. They're doing great.
Where are the bones already? Come on. Come on.
Until we get to what I kind of refer to as the taco phase of the
Taco phase.
the embryo.
Yeah. Okay. Now you're getting into the jargon here. Yeah.
I know that's the real jargon. So they, once you're a ball, the next thing that happens is the front and back of the animal are start to appear. So that happens when like a dent forms in the ball, and you get a second dent, and that becomes essentially the digestive tract eventually. So you have a front bit and a back bit that become the mouth and the anus, and they eventually will develop into all of that. Right now, it's literally like an indent in a ball of cells, and this is not a lot of cells. This is like, if you look at it under a microscope, you see all of the individual little cells kind of sitting next to each other like little beads. So after this goes on for a little while and you have some process there, then there's a thing called the neural tube that depresses again. So you get this long depression that folds up and eventually will close. And that's surrounding everything that's going to go into like your backbone, or at least all of the things that are in the middle of your body along the back. And that is when it looks like a taco because this crack there, this fissure appears in your ball of cells and it starts to get long. So it's slightly long and it's got this little crack at the top and little folds up the sides and it looks like a little hard-shelled taco. Well, that neural tube is developing and eventually the neural tube is going to close. And when you get to that stage with the frogs and the axolotls, then you can start to see some bumps at the front of the embryo that are going to be parts of the brain. And this is taking up a huge part of the body because we're really early in development. So you're looking at like the top third of the animal that's ultimately going to be just a little head on it. But for the time being, it's like a huge part of the embryo of where the brain is developing. So we get our brain, we get some folds and things at the top that are going to become the gills. And then we have, you usually with these guys, with the amphibians, a lot of the things at the front of the animal develop first, and then that tracks down and we get a pattern down the back towards the back of the animal. So you get all these parts of the brain. Eventually, you get kind of like a gut area that develops, and you can see that lump on the embryo that kind of behind these two brain lumps and behind the little gill ridges, you end up with this kind of this gut area. And the part that I'm focused on happens upwards from that, like towards the back from that and behind the brain. There are packages of cells that are referred to as somites. So it is really like a little ball area of cells. And these cells can become they will lead to multiple types of cells. It's because we're still talking about
So they're like stem cells in that sense?
Yes. We're talking about stem cells here. So these are
and you call them somites.
Yeah, so the somite is like a group of stem cells.
Okay.
And when I say it's a ball, like you can look at the embryo and you see just like a nice little round bump. That's the axolotl. The frog embryos are smaller and it can be kind of hard to see them, but they're like a kind of bar shape in the frog embryos and are a little harder to see. The axolotls are much nicer. So on the axolotl, it looks like a nice little bump, and they've got them on both sides. You've got a right one and a left one. And they're on either side of that neural tube, the thing that was the crack in our taco that is now closed over and is kind of a ridge along the back. So these somites form on either side. It's a nice little ball of cells. And within that ball of cells, you have cells that are going to become like dermal skin stuff. You have cells that are going to become, or like at least connective things. You have cells that are going to become muscles, and you have cells that are going to become bones. And in some animals, that section that's going to be bones for you and cartilage is pretty big. In amphibians, that section that is going to become bones and cartilage for you is very small. It's just a tiny little section that's kind of nestled in a particular region of this ball. This is called a somite. So you'd have to get really in there in a way that I have not done to see like these regions of what's going to be muscle versus cartilage and bone. But those are segmented. So you have these balls all the way down the back. The ones in the front appear first. You get your first somite, you get your first like 5 or 6. And eventually like this embryo forms an eye. You still have some somites going on there. In the frogs, they have something called a cement gland. It's what sticks the embryo to a spot once it hatches.
You know what? I always thought scientists were more creative than that. We call it the bone that holds up the skull is called the atlas. Okay, fine. Then cells that glue things together, we call them cement cells. Okay, well, this isn't jargony at all. It all makes sense.
It's so much better when we're not too creative with it. When you get too creative with the names, then it's like, or it's in Latin. It's like, did we really need that? Probably not.
Okay,
not necessary.
We've got the atlas. We've got the cement cells.
And we have the somites down the back.
We have somites. Okay.
And so this is where what exactly is happening with these somites. There are a couple hypotheses of where this is going to go, and it depends on the animal. It's pretty well established for some animals that one somite is going to contribute to 2 vertebrae. And it's not going to make the whole vertebra. It's not just a nice first half 1, second half 2. It's that you have 2 neighboring somites, and the front half of one, the first one, good luck. Where's it go? Where's the first half go? Somewhere in the skull, maybe? Maybe it makes this bone that I looked at in the crocodiles that we don't have anymore, that's in front of the first neck vertebra. Sort it out. Up for debate. But when you get 2 somites next to each other, the back half of one, and then its next little neighbor behind it, the front half of that one, they split and they recombine. It's not really like a clean make 2 balls, then they fuse. It's more like the cells kind of travel to either side. They polarize, they travel to either side, and then they reform to make vertebra. So that's called resegmentation. It's one of the predominant hypotheses of vertebral development, and it's pretty well established in a good number of animals. But then there's some research in, I believe, mice that suggests that maybe resegmentation isn't actually a very good model or a way to understand how their vertebrae are forming. So there are probably different approaches out there. And one of the things that I would like to know is whether this is occurring in frogs and axolotls. And I think it's likely in axolotls. It's not super well known in frogs. I had an undergraduate student I was working with who is on the modern biology side of things. And we were trying to develop a genetic approach to try to visualize resegmentation if it were occurring in frog embryos. Now, like I said earlier, genetics are terrible. No, they're wonderful. They're awesome. They're great for people who are good at them. And I tried. So it's possible that we'll get that project off the ground again at some point, but there's a dissertation to write in my very immediate future. So that one's not moving right now, but I'd really like to know. In the axolotl, what I've done is taken a glowing dye. So when you hit it with a particular wavelength of light, it fluoresces, it glows back to you. And this is the one that I'm using goes and fuses onto the cell membrane. So I've gone and I've put that in a somite. I've said, okay, I think that the atlas of my axolotl is going to be formed by somites 2 and 3. So I'm going to go in and I'm going to, when it's this phase where it has some brain stuff up front, it has this lump that's kind of gut stuff, and it has a couple of the first somites. They haven't reached the tail yet. These things are going to keep going for ages and ages and ages. But I've got my first 4 or 5 somites at least that I can see little lumps on my axolotl. I go in with a tiny glass needle and inject a bit of this glowing dye into that somite. Now, it's going to get in all the cells that are there. It's going to show up in the muscle. It's going to show up in some of these connective tissues, and it's going to show up in the cartilage and the bone. And if I were doing this with a mammal, theoretically, I could get a lot more in the cartilage and bone because there's so many more cells. But I'm doing it in an amphibian, which is highly preferable because the eggs are outside the body of their mom. So I can take them easily without hurting anyone. So I put this glowing dye in that cell, in that clump of cells called the somite, so that it can bind on to their cell membranes. And then every cell that comes from those, so we said they're stem cells, they're going to make a bunch of different kinds of cells. But all of those cells are going to have some of the cell membrane, or at least going to divide out and have a little bit of that dye on them. Or at least some of them will have some of this dye on them, hopefully, so that I can then look much, much later and see where the glow is. So that's happening in this early stage. After that, the somites all the cells in there start to do their jobs. They start to form muscles. They start to form cartilage. And we no longer have nice, clean balls. Those balls are gone, and they're starting to do their job. They're starting to make anatomy. So we get the soft tissue first. The skull is doing a lot of work. There's a lot going on up there, especially if you're a tadpole. Really, all you need to get by as a tadpole is a big mouth and a way to move. And so that is all they are. They're a giant mouth with a tail on it. So they're very focused on the skull for a while, and you start to get all of the bones of the skull that are going to occur starting to form as cartilage. The ones that do form as cartilage first appear up there. And we start to get some patterns. We still have that pattern down the back. We have those segments, and theoretically, resegmentation is occurring. The cells have polarized, and they're getting in their little group. Now, there's a long period of time before the cartilage of the vertebrae appear. So my axolotls need to be around for like 2 months or so that I can theoretically see the beginnings of the bone in their vertebrae. And their skull is like, there's a lot of bone there by that time, but just like everything else, it really kind of starts at the front and then makes its way back, largely. So I need to wait for quite a while to be able to see things that are happening a little farther, even though I'm right behind the skull. I still need to wait quite a while to see bone there. So that's when the animal starts, when these somites disappear, it starts to look more like a little animal. It still doesn't have any limbs or anything, but it does have it starts to form its big gills. They're kind of lumpy and funny for a while, but then they do actually look like gills eventually. And it has its eye. It actually has a mouth eventually. So as an embryo, they're relying on their little yolk sac for a very long time, like way longer than you'd think was reasonable, at least for me. I look at mine and I'm like, shouldn't I be feeding them by now? No. They don't have mouths. I can't feed them. They could not eat if I gave them food. But I spend like probably a good week or two before, or like probably a week before they could eat, looking at them and thinking, I really feel like I should be feeding this animal. And then having to say, no, Dana, they don't have mouths. You cannot feed the animal. So eventually they do look like a tiny, tiny little animal, like, a centimeter long or less, like they're little things. And then they keep their gills, because axolotls are going to keep their gills their whole life. Tadpoles, they're going to have their gills out for quite a while. They're not going to metamorphose for months. So you get these fantastic little things that are a giant head with big old gills on it and cute big eyes. And some amount of tail and body behind. It's a little more regionalized in the axolotl. You can see that they have like a trunk and a tail, whereas the tadpoles kind of just tube out to the tail. And at that point, it's a small animal. And, we've seen tadpoles. You get more and more, you look more and more like an animal. They get some pigment to them, so they start to have dark spots. They have a lot of pigment around their liver. So you see a lot of pigment show up inside them, and then they start to get it on their skin. They have all of their little, the cells that can make melanin start to do so. And they get these spots on them. A lot of our axolotls are the pink variety. So wild axolotls are kind of browny green, and they would have a lot of melanin. But the ones that we have in the lab are these that have been in, captivity for a long time. And there's this kind of long lasting population that are pink, and they don't have a lot of melanin. So you don't get a lot of that in them, but you do see their little mouths. And eventually for the axolotls, their front limb comes first. Their arms start to develop first. And that lasts for a while. They'll have arms. They have like a couple little toes up front before there's even a bump in the back to make its legs. And eventually you get a fully fledged axolotl, which isn't a fully fledged salamander. If it was a salamander that metamorphosed, it would lose its gills and its limbs would probably get a little more robust and it would move around on land. But for the axolotl, we stick it like semi-baby phase, and we have our big gills and we grow our big legs. And
I love the axolotl. I love the creature itself.
Yeah.
I love how it looks. I love how it's spelled. I want to use it in Scrabble one day, but I haven't been able to do that.
Yeah, you need to get like an unfortunate combination of letters that turns out just right.
Yeah, and in fact, I can't show you because it's on the back of my computer, but I have a sticker that says "I axolotl questions."
Love it.
And so
Absolutely.
it's the only sticker I have, but I'm sitting here and I was just vibrating with pleasure that I'm hearing you talk about the axolotl. It's like, I love this little creature.
Yeah, the folks who do develop that do more like genetics in our lab have one of those reversible axolotls where there's a happy side and a mad side, and it sits on top of one of the machines that you use a lot in genetics. And when things are going well, it's happy, and if things go badly, then you'll see the like mad axolotl in the corner of the lab, showing that things have gone awry.
Yeah, look up how they. Anyone that's listening, just look up how these creatures look. I love their appearance.
Oh, they're wonderful. They're
Uh,
absolutely wonderful.
Now, you not only are writing a dissertation, but you've also published a book. Can you tell us a little bit about that?
Absolutely. So, I have a children's book. This happened because I met a 7-year old and she watches the Ontario public television show Dino Dana and could not handle meeting a paleontologist named Dana. She lost her mind. Like, I perhaps it's because I haven't been around a lot of kids for a while. We don't have any kids in my family right now, but I have never seen the like. She flipped out. She was running around the room. She like fell over a few times. I don't know. It was a lot. She was very excited. And I told a friend about this, or I told my partner about it, and he told a friend of his, and she mentioned it to her mom, and her mom is a children's book publisher, and she was like, "Oh, we need to do something with that." So I got reached out to make a kid's book about fossils that was really up to me what it was going to be. So I wanted to write a book about all the cool fossils that are out there. So I said this before we started speaking, but the title is Dana Digs Dinosaurs, and it's a little bit of a bait and switch because the majority of the fossil stuff in the book is about like other cool fossils and like the cool things that we can do with fossils, even the ones that aren't dinosaur bones. Because I think there's just a lot of wonder to be had in all of the like amazing fossils that are out there that you wouldn't necessarily think of if all you've ever seen is just bones, or if all you're ever exposed to is only dinosaurs. You wouldn't necessarily know about the amazing diversity of life that is out there in the world. So I wrote a first draft that was like 100% facts. I was like, here's a fact, and there's a fact, and there's a fact, and there's a fact. And then the publisher was like, okay, Dana, it needs to be a book. Like, it can't be a list of facts. Like,
Right.
so you're gonna, and they asked me to like add a few things at the front that are a little bit about me. So like a little bit about when I was little and how I got into fossils, and then I get to add all my facts.
Yeah. No, no, I mean, you have to tell a story of some kind.
Right.
There's a quotation. I think it might have been Darwin, but I'm not. I forget the attribution, but it's um, a collection a collection of facts is no more a science than a pile of bricks is a house.
Absolutely.
And like and the point is it has to be organized into a structure. Your facts have to be organized into a structure to argue for or against a particular proposition
Mhm.
in order for it to be properly considered a science. And simply listing a bunch of facts uh, are necessary but not sufficient. So
Absolutely.
I'm glad to hear that your publisher pushed back on that. I think that
Yep.
that was a good idea.
Yep. I gave the publisher a pile of bricks, and they were like, "No, no, no. This is how you make a house." And I was like, "Oh, okay, great." And so we did. Sorry, I did a big clack there. We did put together like a little bit of story. So there's a little bit about me, and then there's a bunch of kind of just examples of cool fossils that are out there. A little bit about like how fossils aren't just bones; they can also be from soft tissue, or you can have invertebrate fossils. I know invertebrate fossils don't get all the love, but they're amazing as well. They don't even get all of my love. I'm a vertebrate paleontologist, but I wanted to mention them. And I had a colleague when I was at Virginia Tech who's done some research on color in fossils. So I wanted to mention that there you can even find small structures in fossils, like in fossil feathers, and reconstruct at least some colors that were in extinct animals. So I mentioned some of those things in the book, some of the kind of fossils that I find awesome that I'm hoping that the kids who read the book will also like. They're already into dinosaurs. They already love the idea of these extinct animals, and they can look at it and be like, "Oh, there were also these like really awesome giant extinct fish." And there are also, you can find color in fossil feathers and all of these other really awesome things as well. So that was my goal with the book.
Well, I hope that it inspires a new generation of paleontologists to go out and study the ancient world.
I hope so.
All right. Well, Dana, thank you so much. This has been a great conversation. I appreciate you taking the time and speaking with me today on Podcast for Inquiry.
Ooh, thank you so much for having me.
We hope you enjoyed today's episode. Links for today's discussion can be found in the show notes. If you enjoyed today's podcast, please subscribe and engage in the conversation. Comment, rate, and review. Email us at podcast@centerforinquiry.ca. We'd love to hear your perspective. The Podcast for Inquiry is brought to you by the Centre for Inquiry Canada. We are a national educational charity supporting reason, compassion, and secular values. To our supporters, thank you. If you have not yet contributed, please consider making a donation at centerforinquiry.ca/donate or becoming a member at centerforinquiry.ca/join. Your contribution supports our efforts to have rational and evidence-based decision-making everywhere. CFIC is on the web at centerforinquiry.ca. We are on Facebook, and follow us on Twitter at CFI Canada. Podcast for Inquiry is produced and edited by Lee Shields, Zach Dumont, and Martin Zielinski. Music by Anthony Lazaro. I'm your host, Leslie Rosenblood. See you next time.